Hi there, good morning. I'm going to go very quickly through some work that a number of us, those listed here, both people at Argonne, the University of Chicago, Brookhaven National Lab, and elsewhere, we're doing during our quarantine, targeting, as we say here, SARS-CoV-2 with AI and HPC-enabled lead generation. So the basic idea of this work is we're as part of a large multi-lab collaboration that's seeking to find ways to disrupt the replication and action of the SARS-CoV-2 virus. As we probably all know by now, the virus is a very small little engine that it needs to engage with human cells in order to replicate. We know now that it comprises about 28 unique proteins, some of which are shown here. Science has determined the structure for many of these proteins. So an interesting challenge then is, well, actually two challenges that we've been working on. One is determining more accurate structures for some of those proteins by looking at them at room temperature rather than in cryogenic environments, as has been done before. And secondly, looking to identify small molecules that might be able to dock with key sites in these proteins in order to prevent them engaging in various nefarious activities that they would otherwise engage in. So let me say a little bit about each of those things. They both have involved the use of parcel. So determining structure involves the use of serial X-ray crystallography devices at Argonne's advanced photon source. Identifying molecules involves the use of machine learning, basically, to screen very large numbers of small molecules to find those that might have interesting properties. Serial crystallography, serial synchrotron crystallography is a way of, it's like crystallography, but it basically involves not performing crystallography, X-ray imaging on a single crystal, single large crystal of a protein, but many small protein crystals, which allows for imaging of devices of proteins that could not otherwise be easily characterized. That's the advanced photon source on the top right, and the bottom right is a chip which has many thousands of embedded protein crystals. So Ryan Chard on our team worked closely with people at the advanced photon source. Oops, we've even got a little molecule here. This is pretty amazing. This is zipping around at very high speeds, collecting data. We implemented a data collection, analysis, quality control, metadata extraction, data storage and indexing and storage pipeline using a tool called Globus Automate. With under the covers, parcel being used for data to perform high performance data analysis, reconstruction, and so forth. And I won't spend a lot of time on that because, of course, time is limited, but let me just move on. So the one output of this work was a set of new structures of COVID proteins obtained, as I sort of mentioned earlier, at room temperature rather than in cryogenic forms. And if you can see the middle here, this shows some of those structures available on the Globus-based Petrel data store that we've developed and that we use at Argonne for many advanced photon source data. And the key point here was, on the parcel side and the Globus Automate side, is we were able to provide them real-time feedback on the quality of the data they were collecting by performing reconstruction as experiments were happening. And that really made a big difference to how that work was done. So the second activity that we were engaged in was screening candidates. So we've got some proteins, we know something about their structure and places on the surface of the protein that we'd like to interfere with by having a small molecule bind to it, preventing it from doing other things. The challenge is finding such small molecules. So there are essentially infinite number of potential small molecules. We have databases of 4 billion drug-like molecules for which people have determined various aspects of their structure. Obviously, we're not going to be able to take 4 billion of these small drug-like molecules and test them all in clinical trials. We can't even test them in a wet lab. That would be totally impractical. In fact, even performing high-resolution protein docking simulations for all of these proteins would be impractical. So instead, what we do is we first of all perform high-throughput computational screening, computing for all of these 4 billion proteins, the various fingerprints and other properties. And then we use a combination of previously performed protein docking simulations and machine learning methods to build predictive models for various properties such as docking likelihood. Other things might also be of interest. And that's where we made extensive use of Parcel over a period of several or many weeks, building large collections of these databases, performing high throughput canonicalization to come up with a standard structure, computing a whole range of features, computing fingerprints, generating images, and then performing various other computations like similarity search, deep learning, filtering, and so forth. And this ultimately this used a whole series of computer systems across the US, at Argonne, also at TAC in Texas, and other systems that we had access to. And the final output was, well, I shouldn't say the final output. The output so far has been tens of terabytes of computed properties. These themselves have been used to to train machine learning models that have themselves been used to characterize these 4 billion or so molecules. And at this point, we've got several hundred candidates that are being synthesized and tested in the lab. So we have hopes that this will lead to interesting outputs from the perspective of COVID therapeutics. So thank you very much. I'll be happy to take questions.
Wonderful and perfectly timed. Thank you, Ian. Any questions? We've got a couple of minutes. Questions from the crowd.
I would say Parcel was cool. It was great. We're using it on all sorts of systems. It was nice having Yadu and others to help us, but really it's allowed us to do some amazing things.
We've paid you to say that, I'm sure. With the SSX stuff, Ian, so the idea there is it time sensitive? Is that feedback loop you showed with the nice arrow there, is that something that needs to run very quickly, or is that an asynchronous process that can go and run quite slowly?
No, it's fairly time sensitive in that the scientists want to know whether to continue data collection or abandon a particular protein, because they generate these samples and their quality varies significantly. So that's one reason for wanting real-time input. Another reason may be that in one of these arrays you have thousands of samples. You may determine after a certain amount of time that you've got enough data to obtain a good quality reconstruction, and then you can move on to the next. And of course, they only have very limited time at the X-ray source beamline, so anything that can either avoid imaging things that are no good or stop imaging things that are good and move on to the next thing is a great saving and helps move the science forward.
That's awesome. I guess that's useful for any of these type of experimental, instrument-driven science. So maybe the work Matthew does even would be interesting there. Okay, well, thank you very much.